Meiosis. So now that we've discussed mitosis and the cell cycle, we're ready to go to a specialized type of cell division that allows for great diversity, and it's the way that organisms have moved in order to do sexual reproduction. So without further ado, I'm not going to look at these, but you may want to go back to the Prezi later on and look at these different things that you should be expected to do after we finish this chapter. So, first thing, before we even start, whoops, wrong way. Here we go. Before we even start going into sexual reproduction, let's just sort of get out of the way the fact that some, even multicellular organisms, can reproduce by asexual reproduction, basically making a clone of themselves. Here is a tiny aquatic organism, a hydra, that literally grows a bud from the parent. This will be a clone, it will be identical cell, organism, and that bud's off and you can make a new one. It doesn't allow for much diversity except for mutation that takes place. California redwoods, if anybody's gone hiking, you've seen, I hope, the fairy ring that happens. So in the center was the old parent tree, and then around it can grow up its clones or its offspring. And then it's near where the parent was. It's basically a clone of the parent. Again, that doesn't allow for genetic diversity and may not be so good for survival. This is a picture. We've talked about chromosomes in the last chapter, but now is a chance to talk about it again. And these are real chromosomes. So we can purify real human chromosomes and mark them, maybe if they're going to do genetic analysis, to make sure that all of them are there. It also is a great way to display to us that there are 23 unique pairs of chromosomes. 1 through 22 are ordered by size, with 1 being the largest and 22 being the teeniest. It was the first one that was completely sequenced. And then X and Y, this would be a male. They are the 23rd pair. If you're a male, you have 2 different sized ones. If you're female, you're XX. So now getting into it a little bit deeper, let's think about what it means to have pairs of chromosomes. So we're going to hear this N term a fair bit amount. So that would be in humans the 23 case. So we have 23 pairs, so we have a total of 46. In this key that they're showing you here, we're just going to look at 3 sets of chromosomes. So N is going to be 3. You get half of your genome from your mom, maternal, and half from your father, paternal. Remember, before meiosis commences, just as when we talked about the cell cycle before mitosis commenced, you have to duplicate your chromosomes. So the duplicated chromosomes are called sister chromatid. They're held together at that centromere. There you go. And in mitosis, they did not need to find each other. So if this was chromosome number 1, the large one, did not need to find the other homologue of chromosome number 1. This might be chromosome number 2, the mid-sized one, and chromosome number 3, just for our picture purposes. But in meiosis, they are going to have to find each other because some interesting genetic diversity events are going to take place. Okay, so I think we have our nomenclature down for now. Sorry, that cut off. But almost all sexual reproduction involves the same cellular process. You have one haploid cell, meaning half of the genome, 1N, meeting up with another haploid cell, 1N, to make a diploid cell, which is now 2N. So haploid is half of the genome, diploid is the double copy. Again, in humans, we have 23 pairs of chromosomes. We have 2N of 46. Fruit flies are different. They have only 4 chromosomes, so their 2N is 8. Dogs, 39. 2N for them is 78. Different species have different numbers. It's one of the reasons, one of the ways we define a species. Come back to that in evolution about February time. So then I just wanted to direct your attention to the life cycle of humans, since we're most likely to identify with that, right? An egg, which is haploid, gets together with a sperm, which is haploid, upon fertilization to make a diploid zygote. Sorry for all the vocab, but you definitely want to know these words that I'm going over right now. That zygote goes through mitosis and development to make a full-formed organism. And then at some point, only in the gametes, we produce haploid cells again. So in females, the ovaries are where the gametes are produced, and in males, the testes are where the gametes are produced. About April, we'll go into those details, but right now you know that you make a haploid cell, which goes through meiosis, which is what we'll talk about today, and then they can come together to make a new organism that's diploid. This figure always upsets people, but it's actually not so hard if we look at the color coding. And the point is that if animals are some of the most sophisticated, advanced, multicellular creatures, we can see that we actually spend a large chunk of our conspicuous life cycle as diploid. The mouse, humans, frogs, everybody, right? And then only the gametes that are be involved in reproduction have a haploid. Now, as you kind of move down the diversity scale, and you look at things that evolved earlier than us, you'll see that some plants, early plants, actually spend a pretty conspicuous time as actually a multicellular haploid creature. Moss is a great example of this. We'll talk about it later on in the year. And the fuzzy green stuff that you see growing around is actually haploid, and it's only after eggs and sperm are made that they join together to make a zygote, which then undergoes mitosis and then quickly makes the stage that you really see. Then if you go even further down to earlier in evolution, some things that represent the fungus and the protista, single-celled eukaryotes and multicellular that are not animals and not plants, they actually spend a fairly short or very inconspicuous time as a diploid thing and spend a lot of time as haploid. That's all I really want to talk about for that. We're going to focus the rest of the time on basically humans. This is a very detailed summary comparing mitosis and meiosis. When DNA takes place, both of them are before the whole process of dividing the nucleus goes on and dividing the chromosomes. The number of divisions, mitosis is just once. We're going to see meiosis goes through 2 times to make 4 cells instead of just 2 cells. I want to talk about synapsis and crossing over as we go through. It doesn't happen in mitosis. It will happen in meiosis. And then the rest of this I'm going to let you read because I think it's just a whole lot of words that I want to talk about right now. So let's start talking about what's going to happen. So interphase in meiosis would be before we are able to divide. We have to have chromosomes duplicating, and then they start to condense once we approach meiosis 1. So those duplicated chromosomes are going to be separated. So this is just chromosome number 1. Got one from dad, one from mom. I'm going to go with the blue ones for dad and red ones for mom. Then they get duplicated during some S phase. And then we're going to have them close together. And then at the first split of meiosis 1, the homologous pairs separate. At that point, at the end of meiosis 1, we technically have a haploid cell because you have half of the genome. Then there's another round, meiosis 2, where those cells separate again, and we end up having 4 unique cells, and you'll see how unique they are at the end. Let's look at it kind of in a timeline here. So this would be the beginning. Meiosis 1 is when we're going to make that haploid thing and separate the homologous chromosomes. So prophase 1 is the busy action time, just like we had in prophase of mitosis. The sister chromatid are going to be together at the centromere. But the interesting thing that happens here is that they must pair up with their homologous pair. So they make what's called a tetrad, 4. This chiasmata forms, which is basically an area of overlap of homologous chromosomes, and we'll see that in a little bit closer detail later. Again, similar to prophase and mitosis, the nuclear envelope starts to degrade, and we have the 2 centrosomes with the spindle attachments forming. Then we move on to metaphase, where those chromosomes again line up on this imaginary plate in the center. And take note of the fact that the red ones, those sister chromatids, so that's chromosome 1, 2, and 3 for our purposes here, they don't always line up with the maternal ones on the same side. So they kind of mix and match. Every single pair of chromosomes, so for chromosome 1, 2, 3, all the way up to 23, has a 50-50 chance of putting their homologous pairs on either side. This is a huge location for, or opportunity for diversity, because there are 2 to the 23rd, and I'll go over this again, different ways you can line up on the metaphase plate to make a unique cell that's going to be at the end. So again, this is a big important time during meiosis where those kinetochore microtubules attach, and they will then begin to pull things apart in anaphase 1. We'll say it over like many times. Sister chromatids stay together, and you know they're sisters because at that centromere we have 2, in this case, blue chromosomes attached to each other. These used to be identical. Note there's some new stuff here, and we're going to go into some pictures on how that happens. We have crossing over that takes place that introduces some diversity. Then we've got telophase and cytokinesis, where we literally rebuild our nuclear envelope or start to rebuild. Usually it doesn't completely form. We have the cleavage furrow, and then we'll have 2 cells that at this point are not haploid. Then those 2 cells will then undergo basically the same process. We have to have our duplicated centrosomes, our microtubules form in prophase 2. We have all the chromosomes line up on the metaphase plate in metaphase 2, and in this time the sister chromatid will be separated from each other, and we will have 2 completely haploid cells with very unique compositions in there. Note that this process of making 4 cells happens in sperm in humans. If we were making eggs in humans, we actually never make 4 eggs. We actually donate all of the cytoplasm to 1 cell and sort of throw away the excess chromosomes so that only 1 haploid cell is preserved in an egg. Don't worry yourself about that right now. We'll talk about it again in April. So, again, we can compare and contrast the mitosis and the meiosis. I don't want to dwell on that too much, except that we know that mitosis's goal is to make 2 identical cells. They are diploid, and they should be identical. If they're not identical, then a mistake has happened, and usually that's not so good for a cell. However, in meiosis, our goal is to make a very unique group of cells so that we can reproduce and share diversity. So if you look at it down here, we have a solid red chromosome, we have a red chromosome with a blue tip, we have a solid blue chromosome, and now we have a blue chromosome with a red tip. So if we're calling this chromosome number 1, it has 4 completely unique chromosomes that did not exist in the parent, mixed up with other guys here. So let's go on and talk about how that uniqueness takes place. One major way to get uniqueness is independent assortment. And I mentioned this before. So crossing over, that I've talked about a couple of times, happens all the time, and it can even happen multiple times on the same chromosome. And it happens because these homologous chromosomes actually code for very similar information. Their DNA code, their GATC, GG, blah, 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 is very similar because they're coding for the same genes. We talked about this, and we'll continue to talk about this, I hope. So they are attracted to each other because DNA likes to hydrogen bond. There are enzymes involved in this process, and this actually they've seen it happening in real chromosomes. This crossing over point is called the chiasmata, and basically enzymes come in and literally break apart the chromosomes and reform them this way. So note here, only those inside chromosomes cross over, never the outside ones in this round. And they are more likely to be crossing over at the tips. They are very less likely to be crossing over at the centromere, mostly because there's proteins in the way and it's just not as flexible and easy to have the proteins come in and hold it together. Sometimes they can cross over a couple times. So then in anaphase 1, the blue chromosomes that are tethered together, so the blue sister chromosomes stick together, sister chromatids stick together and go to one side here. In this case, the red sister chromatid are going to go to this side. And then in anaphase 2, when we completely separate the sisters, we now have them each in the haploid cells, a full blue one, a mix and matched, a mix and matched, and a full red one. Just a closer up picture of the chiasmata. So, just to remind you, the different places that we have diversity taking place. So, crossing over happens in prophase 1. The homologous pairs find each other, cross over. Metaphase 1, this is what I think that other figure was actually not doing a very good job of. Each of these 3 chromosomes has a 50-50 chance of lining up on the metaphase plate this way. In this case, the red one ended up on the right. The paternal ones, or the blue ones, ended up on the left. In the case of number 2 here, we've got the blue ones on the right, red ones on the left. This one, blue ones ended up on the right, red ones on the left. 50-50 chance. Since we have 23 chromosomes, we have 2, because there's a 50-50 chance, 2 different ways. 2 to the 23rd different ways that this can happen. And that's what I have here. So independent assortment can result in 2 to the 23rd different ways for homologous chromosomes can separate. That ends up being 8.3 million different ways each time you undergo meiosis. Crossing over occurs on 22 of the 23 sets of chromosomes, which allows for a mixing and matching. And then lastly, the last opportunity for diversity with sexual reproduction is that the dad sperm is hopefully genetically unique from mom's egg, and now they're going to fuse to become a new person. So it is actually quite amazing that siblings ever look alike when you think about all the different ways that you can have a unique egg or sperm made and come together. And then lastly, I just wanted to remind you that if you go to the original one, this is the Bozeman biology. I always love the way Paul explains what I just explained in his own way with different pictures. So, oops, didn't want to hit that. By all means, please go and take a look at that.